Okay, group, this is our first set of notes for our next unit, which is over genetics and inheritance. So let's go ahead and jump into it. All right, so very first thing, just going through and defining a couple of terms or a couple of things that we really need to know. The first one is just answering the question, what is inheritance? To inherit something means that you receive it from biological mom and biological dad. For the sake of this unit, I will refer to parents as mom and dad, but just remember that is strictly for vernacular and just saying that like the traits were passed on from quote mom and dad. But we know that mom and dad is sometimes something a little bit more than who you received your traits from. But we will speak in terms of human traits, but we will also speak in terms of like animal traits. And so just saying mom and dad is faster than saying biological mom, biological dad, or the biological maternal trait giver versus the biological paternal trait giver. So we'll just say mom and dad. So inheritance is when you pass on the traits to offspring. So mom and dad pass their traits on. Um, the biological basis for inheritance, so how all of this happens, how these things are passed on from parentals to offspring, is done by genes. So we've heard the word genetics, we've talked about DNA. So to be more specific, a gene is a like chunk or a segment of the DNA that codes for a particular set of proteins that make something happen in the organism. So again, we've talked about genes for like eye color, eye size, eye shape, whether or not your optic nerve works, whether or not you even have eyes. All of these things are coded in some way, shape, or form on a gene on the chromosome, and then you get half of your genes from mom, half of your genes from dad. And that is what inheritance are, which is why offspring will frequently have those similar traits that mom and dad have. Okay, so if we think back to meiosis, and we've done the review activity just to kind of refresh our brains on what meiosis is. Meiosis makes it so that when an organism is getting ready to make their gametes or their sex cells, it creates a little bit of genetic diversity. And it does that via crossing over, which happens during prophase 1. So if we look here, we've got our homologous chromosomes. Again, that just means they are the same chromosome. So this would be dad's chromosome 1 and dad's chromosome 2 for whatever it is that's coding. The crossing over occurs where technically this would be grandpa's chromosome and this would be grandma's chromosome because dad got half of his chromosomes from his dad, and then half of his chromosomes from his mom. So now we're mixing and matching basically grandma and grandpa's DNA in order to get dad's gametes to be a little bit different from each other. So again, that's something we have reviewed in class, and then we will continue to talk about it just because using mom and dad and grandma and grandpa can get kind of confusing. But key thing is that diversity happens because of this crossing over that occurs during prophase 1. And then once we go through the rest of meiosis 1 and then the rest of meiosis 2, that is when we get the four separate gametes. So that would mean that this particular chromosome here, it would split. So this would be, come on, why is my pen not working? There we go. This would be gamete 1. This would be gamete 2. So notice how gamete 2 has a little bit of dad's mom's DNA on it. Gamete 1 has all of dad's dad. And then we could then flip over to gamete 3, which is mostly of dad's mom with a little bit of dad's dad. and then gametes 4, which is all of dad's mom, so grandma. So again, kind of weird, it gets a little confusing, but we will hopefully go over it some more so that those confusions are gone. But because every single one of these chromosomes then that will be into a single gamete are different, it will make it so that the 4 potential offspring will also be different. So, and then you have to remember that like in humans, this happens 23 different times. Like each gamete has 23 different chromosomes in it. So you have a lot of opportunity for diversity and variations within the genetic code. Okay. Now, sometimes things do not go according to plan. We've learned that with like mutations, things can get messy. And one of the chromosomal disorders that can occur, so one of the things that can happen that's not necessarily a mutation, this means that during meiosis, so not during DNA coding, but during meiosis, a mistake occurs. So this first one is called a nondisjunction. And during a nondisjunction, instead of if we have this original cell and it's going through meiosis, instead of everything splitting equally down the middle during meiosis 1 and then doing it again during meiosis 2, resulting in each cell having equal amounts of chromosomes, during non-disjunction, the chromosomes don't separate properly, and so now you have abnormal numbers of the chromosomes. So in this particular example, the gametes were supposed to have one big chromosome and one little chromosome. Okay, on here the colors are red and yellow. But oh, trying to get my markers back. There we go. So we have, let's see if we can zoom in. Nope. Yeah, there we go. Okay, so we have ideally, theoretically, in a perfect world, each gamete would have half of one of these chromosomes, this top chromosome, and then half of one of these bottom chromosomes. Well, non-disjunction means that you have either too many, as is the case on these left side gametes, or too few, as is the case with this right side gamete. So with these abnormal numbers, if these particular gametes were the ones that fertilize or to make the baby, so remember this would be sperm or egg cells, if this happens and one of these particular gametes was the one to create the offspring, now the offspring has a non-typical number of chromosome, which could then lead to a disorder. An example of that would be Down syndrome. Okay, so yeah, here's just another picture of that. So here is our typical cell that is supposed to be going through meiosis. In this case, this would be like a pre-egg cell or a pre-sperm cell. It goes through meiosis 1, and during meiosis 1, there is a mistake. So this top portion's showing the mistake during meiosis 1, where instead of separating each side to these top and bottom red and blue chromosomes, the big chromosomes stay stuck together during meiosis 1. And then during meiosis 2, everything continues to kind of separate the same way that it would typically. And as a result, you have 2 gametes that have one too many chromosomes in it. And then you have 2 gametes that have one too few chromosomes in it. Now, non-disjunction can also happen during meiosis 2. So again, during meiosis 1, everything separates typically. Then during meiosis 2, when the chromosomes realign down the middle, the one or a set of the chromosomes don't split at the centromere, and as a result, the chromatids don't separate. So this would be the case here in this top cell. So during meiosis 2, you now have one gamete that has too many you have one gamete that doesn't have enough chromosomes. And then this bottom cell went through meiosis typically. And so they have the correct number of chromosomes, which is represented by the letter N. Now, we've already talked about chromosomal errors, where this happens via an actual mutation. So we have deletions and partial deletions and duplications, excuse me. And that's just where part of the chromosome. So again, the chapter of the book is multiplied, it's written twice, or it is completely cut out. We have inversions where the gene flip flop, the chapters flip flop. So you're reading your book and you get to the end of chapter 1, and then all of a sudden you are reading chapter 14 and you have mixed and matched everything. And so now the location is different. And then translocation, this is when the where you have part of one chromosome separates and then goes and attaches itself to another part of a completely different chromosome. And that's what we have down here, the reciprocal translocation. So here we have uh, this part of the big chromosome and this part of the little chromosome. They flip flop. And as a result, again, you're not gaining or losing any information in both inversions and translocations, but now it's out of order. And genes really like to code in a specific way and in a specific order. So if you're messing with that order, it can cause issues. So a few more kind of vocabulary terms. The human genome itself, that means it is the entire set of organismal DNA. There is the human genome. Again, this started all the way back in the 70s. During the 80s, it was really just a time of trying to figure out how can we get this code done. And you have to remember that this was during a time that there was no internet, there was no way to get information back and forth really quickly. So the 80s and the early 90s were really just spent like trying to figure out how do we get these machines, these computers, which again are fresh off the market, how do we get them to code quickly and efficiently, and how do we talk to all of the other groups that are doing the same project. Now, mid to late 90s, computers start getting faster. And then the early 2000s, things really take off. And by, I want to say it was like 2010, 2008, something like that, the full human genome was complete. So, anyways, kind of a tangent off of that. But so a genome just means it's all of the DNA, it's every nucleotide being read for that particular organism. So the genome is going to start with chromosomes. We have to look at those bundles of DNA wrapped around those proteins called histones. And then we photograph it during mitosis, and then you condense them to be able to view them. And taking all of this information, taking the DNA and photographing it and looking at what it looks like when it's fully condensed during mitosis, because remember, the DNA is only condensed during mitosis. While that's happening, and we make a little picture out of it, we call that picture a karyotype. So you've probably seen something like this before where you have all of and I'll go back to the last slide, so don't panic if you missed it. But you have like all of the different, in this particular case, you have all of these different chromosomes for the human. And these are your homologous structures. Okay. So if this, for example, if this was my DNA, then you would be looking at one of the chromosomes comes from my biological mom, and then one of the chromosomes comes from my biological dad. And then remember, during meiosis and prophase 1, that crossing over happens, and then the gametes get passed on to offspring. So my children technically have information from my parents, but that's how inheritance works. Karyotypes just make this so it is visual, and you can if you go to Google and you type in karyotype, you're going to have hundreds upon hundreds of images pop up that are all showing the same thing, but different because it depends on the organism. But we can see here in this particular karyotype that a human karyotype or a human's full set of DNA, or excuse me, full set of chromosomes has 23 sets, so 46 chromatids, all of that fun vocab. So here we're back at that previous slide. When we look at those photographs, we put them together, we make them nice and pretty. We put the homologous structures together, and then we call it a karyotype. So that karyotype is looking at the diploids. Remember, diploid means that it is both chromatids grouped together, and they are also done so by size. So again, if we look at this, we can see that chromosome 1, so it's a diploid because it contains 2 sets, but this is the biggest chromosome. Chromosome 22 is the smallest. And the 23rd chromosome, this is our sex chromosome. So this is the one that's responsible for determining the sex of the organism, male versus female. And we'll get into a little bit more of the biology behind that, because sometimes it's not so simple as XX or XY, which is kind of interesting. Okay. So as mentioned, a human cell, each human cell has 46 chromosomes or 23 pairs of chromosomes in each cell. And again, it determines many things. It determines the sex, if there's any extra or missing chromosomes, any chromosomal abnormalities, any mutations will be coded in here as well. Oh my gosh. Okay. And so that's what we can look at in this particular picture, is we're looking at this. Okay, this would be a typical autosomal, for the most part, autosomal karyotype. So chromosomes 1 through 22 are our autosomes, and then our sex chromosomes are not autosomes. Autosomes just basically means that it is true homologous structures, and that each one is coded pretty much the same way. Whereas our sex chromosomes, if you look, if you have an XX, which means biologically the organism is female, those 2 chromosomes are homologous. But if the organism is male, XY, the X chromosome is bigger than the Y chromosome. So that's why the sex chromosomes don't get to be considered autosomes. That is why they are considered just sex chromosomes. Sex determination. Again, this is when we are talking about sex. This is talking about the that 23rd chromosome. We are not talking about gender, 2 completely different things here. So when we're talking about sex determination, we are talking about the chromosomes in the organism. If the organism has an X chromosome, which basically means a big chromosome, and a Y chromosome, which is little, that means they are male. And if they have 2 big chromosomes there, 2 X chromosomes, that means that they are female. This is, again, strictly genetic. This is strictly inheritance, because some organisms are able to flip-flop their sex based off environmental conditions. So an example of that would be temperature in reptiles. Some reptiles can change their sex depending on the temperature and the climate outside. And another example would be clownfish. So in the event that a male and female companionship between clownfish, if the female dies, the male will actually switch his gender to female so that he can lay eggs. So everything you know about Finding Nemo and Nemo having a dad for that long is actually scientifically a little inaccurate, because Nemo's dad, Marlin, should have switched to female so that he could lay more eggs. But kind of interesting there. So just kind of comparing the size of the X chromosome versus the Y chromosome. So the X chromosome codes for like over 1,200 genes. That's why it's so big. Whereas the Y chromosome only codes for 140 genes. So it's a lot smaller. And then mostly it is genes associated with like male sex determination. and sperm development. So on this Y chromosome, that's where you have testes, like where the testes form, the formation of the penis, and then you also have the formation of sperm. And then any association of like genetic conditions that are more specific to males. So, like male pattern baldness, if that's something that runs in your family, that's found on your Y chromosome. Interestingly enough, as well, muscular dystrophy is an X chromosome disorder. So, that's where your muscles will over time start to, uh, for lack of a better term, fall apart. They deteriorate over time. Hemophilia, which is a blood condition, is found on the X chromosome, and color blindness is also found on the X chromosome. And that is it for this first set of notes. If you have any questions, please let me know. Otherwise, we will discuss and do some activities in class with this. But as always, answer your questions, and I will see you in class.